All right, everyone, welcome to our first histology seminar. It is on the topic epithelium and glandular tissue. My name is Ria, and me, along with Varun and Catherine, we'll be Katerina. Sorry, my bad. We will be presenting it together. All right, Varun, you can start.
All right, hello guys. Before we begin with the topic, I wanted to highlight one important thing. So the topics that you will be doing in these first 5 weeks, that is the epithelium, connective tissue, muscle, nervous tissue. These will form your basics for the rest of the year. So this information is what you need to know and like throughout the year because all these characteristics will be present in all the slides. So this is important for your slide test, for your micro test, or even in class. Professors can ask you this anytime. So what my advice or like what our advice would be that you guys study these 5 topics at least in a way that you can retain at least some information all the time because you have to come back to this information again and again because you can be asked about this at any exam or any test, even in your obviously in your final exams as well. So let's begin. Uh, so I would like to ask some questions during the presentation. So it's up to you guys if you want to answer in chat or you want to just answer, you can unmute yourself. Up to you guys. So let's begin. What are tissues, people? No response. Guys, I need some interaction, so I know someone who I'm talking to. Otherwise, I'm just talking to my laptop. That's not.
Tissues are a group of cells that have a similar structure and function.
Mm-hmm. Yeah, that's it. So yeah, tissues are a group of cells that all look the same and carry out the same function. And here it says tissues, organs, and organ systems. So basically, the thing is that our tissues, they are like team players, So they don't just like sit around and do nothing. They come together, they form organs, and these organs like come together forming organ systems. So think of it as like, tissues are building blocks of the house, and the house is your organs, and then these organs come together to form your organ systems. Now, does someone know any types of tissues?
Bone tissue.
Yeah, but I'm talking about like, general classification of the types of tissue. You said
Connective
specific.
tissue?
Yeah. Anything else?
Connective, epithelial, nervous, and muscle tissues. 4 types.
Exactly. So yeah, types of tissues would be the epithelial tissue, connective tissue, nervous tissue, as well as the muscle tissue. So let's begin with the epithelial tissues. So the main gist of is that the epithelial tissues, they form your body covering. So as it's written there, they form the protective barriers on the body surface, and they help in certain processes such as protection, secretion, as well as absorption. Now they're made up of cells which are tightly stuck to each other with some extracellular matrix between them. Now you guys must be thinking, what is extracellular matrix? Extracellular matrix is basically, like soft stuff which fills the spaces between the cells, and it like helps the cells, it supports the structure of the cells, and it helps things stay in place. Without it, everything would just like fall apart. Now, the cells, which the epithelial tissues, they are separated from the underlying tissues by the basal lamina. So my Ria would be explaining you further about basal lamina later, but the main idea about basal lamina is that it's like floor which separates the cells from the underlying connective tissue. So anything that has to pass from like the connective tissue to the cells has to go through this basal lamina. That's it. It acts like a filter. So, yeah, that was epithelial tissues. Now we'll talk about connective tissue and supportive tissue. So, the connective tissue, it provides structural support and stability for the body. It is made up with cells with large intercellular spaces and it contains extracellular matrix. Again, extracellular matrix is the soft stuff that maintains the structure of the cell. And then we'll talk about the nervous tissues. So nervous tissue, I think most of you guys already know, it transmits electrical signals throughout the body and it allows communication between brain, spinal cord, and rest of the organs. Like it helps activating specific muscles as well as glands. It is made up of cells that accept stimuli, conduct impulses, and process information. Now, another point to remember is that they have supporting cells called neuroglial cells. So now what neuroglial cells are, they act like, they act like bodyguards of the neurons. So what that means is that they do not like transmit or send some information. What they do is that they make sure that the neurons, they are well fed, any damage to them is like repaired, and and they make sure that the neurons carry out their function properly. So just like think of it as like a supporting function for the neurons. Now, the next one would be the blood. Now, the blood is also a type of liquid connective tissue and it is made up of fluid matrix called plasma. So what we basically mean by that is the fluid matrix, that plasma is like the watery component of the blood. So blood has two components. It has cells and then the watery component under which plasma comes. So the cells would include erythrocytes, leukocytes, lymphocytes, all that stuff. And then we would have the plasma, which was the watery stuff. And it is more in quantity compared to the cellular components. So just to summarize the slide, connective and supportive tissue provides structural support and stability for the body, large intercellular spaces, extracellular matrix. Nervous tissue transmits electrical signals, basically controls your whole body, communication between cells and stuff like that. And most importantly, have neuroglial cells that support them and protect them. Now, next would be the muscle tissue. You will have a specific special micro test for this. So just for now, general information, it is specialized for contraction and responsible for producing movement inside the body. Now, characteristics of the epithelial tissues. Do you guys know any characteristics of the epithelial tissues? I mean, you can see in the picture, but still.
They have intercellular junctions.
Mm-hmm. Anything else?
They might have surface specializations like cilia.
Mm-hmm.
They're
Yeah,
avascular.
right. All right. So characteristics of the epithelial tissues. These are the four main things you need to know. Intercellular junctions, polarized morphology, basal lamina, avascularity. So intercellular junctions, as I said before, the cells are tightly packed together via these intercellular junctions. So it allows these cells to stay together to prevent the breaking of the structure apart. Then the polarized morphology. So, I mean, it might sound like a complicated term, but it basically means that epithelial cells, they have two sides. One is the apical side, as you can see on the diagram. One is the apical side, the other is the basal side. The apical one is the top one, so it would be involved in things like absorption, secretion, and the bottom one would be like, the supporting part. It helps it keep stuck to the basal lamina. So it's like, the epithelial cell would be a coin which has two sides, the top part and the bottom part. You can think of it like that. Then, as I said about the basal lamina, the cells are separated from the underlying connective tissue by the basal lamina, and the underlying connective tissue would supply the cells lying over it with like nutrients and stuff like that. So the underlying connective tissue underneath would act like delivery system, food delivery system that would provide nutrition to the cells above. Now, the last point would be about the avascularity. So basically, this means is that these cells, they lack blood vessels, meaning the nutrients have to diffuse from the underlying connective tissues. Like I said, since the cells above, they do not have their own blood supply, this is why the nutrients have to diffuse from the underlying connective tissue through the basal lamina. But the only exception about this is the stria vascularis, which is present in your ear, which contains its own blood capillaries. So once again, just look at the diagram. You have the intercellular junctions, polarized morphology, two different ends, apical part, basal part, then the basal lamina, nothing but a float which separates from the underlying connective tissue, the above cells, and avascularity. Now, uh, yeah. So what do you guys think? How can we classify epithelial cells according to their construction? Like what would be the
Trabecular.
general, yeah, say?
Trabecular.
Hmm?
Trabecular?
Uh-huh. And
Surface and reticular, I think.
yeah, yeah, that's it. So there are three types, three ways in which we can classify this information. So one would be flat, trabecular, and then reticular epithelium. So now about surface flat epithelium, it will form a continuous layer lining on the inner and the outer surfaces of the body, and this includes simple or stratified layers. So basically what this means is that simple would be like one layer of cells, stratified layer would be like multiple layers of cells. So yeah, that's about it about this. And then we have the trabecular epithelium. So the cells in this case, they form interconnected plates that create a three-dimensional network, and the arrangement of this epithelium facilitates interaction between the cells. So what does this interconnected plates mean? So it basically means like, the trabecular epithelium, its different parts are like, a puzzle. So different plates and or like layers, they fit together and they come together and then they form this type of epithelium. Like it is seen in organs like liver. And then we have the reticular epithelium. So it forms a network of star-shaped cells connected with each other and it forms a supportive mesh. So as you can see on the diagram here, you can see these are the stellate-shaped cells and they're sending out, a long processes that allow them to connect with each other. It's like a spider web. So this is how these cells are connected and they form this epithelium. So again, classification according to the construction, surface flat epithelium, continuous layer, two types, simple or stratified. Then we have the trabecular epithelium, interconnected plates or layers that come together to form a 3D structure. Then reticular epithelium, star-shaped cells connected by their processes or arms, whatever you want to call it, to form a supportive mesh. Then we're talking about the classification of the epithelial cells according to function. So does anyone have any idea about this?
I think it's covering, and the thing is glandular, in
Yeah,
case if that's
exactly.
for the test.
Mm. So the three classifications would be the covering epithelium, glandular epithelium, and special epithelium. So covering epithelium, as I said before, it forms the outer layers of the organs and body surfaces. So it's like a body's coat and it covers and protects your organs from the outside world. So it basically acts as a barrier. That's it. Then we have the glandular epithelium. So it is specialized for secretion, forming glands, and it is involved in release of hormones as well as digestive enzymes. So it's like a factory inside your body which releases hormones basically. And then you have the special epithelium. It includes various types with special roles, such as sensory epithelium in the ear and the nose, and others like respiratory or pigment epithelium, which forms specific tasks inside the body. So that was about the function. You have the covering epithelium, that is the barrier, glandular epithelium, which releases special secretions, and special epithelium. It can be like, a multitasker which can carry out specific tasks related to their like function or like location. So yeah, that was. So Ria, you can take from over here. Yeah.
Just a second.
While that's happening, does anyone have any sort of questions regarding Varun's segment, what we just did? If you have, please just feel free to ask. If not, Ria is just going to start.
All right, okay. So, like Varun said earlier, covering epithelium is the epithelium which covers the entire all of your body surfaces and even your internal organs and everything else. So, does anyone know how to distinguish between the different types of epithelium based on this diagram, maybe? Like, what are the types of epithelium according to the number of cell layers? Does anyone know?
Louis?
You have simple and right.
Yes, exactly. So simple and stratified are the two main things which you can say depending upon the number of the cell layers. So simple epithelium basically means that there's only like a single layer of cells which you can see. But stratified means that there's going to be multiple layers. And pseudostratified is another thing. So this basically means that they're completely like different irregular shapes of cells, and the nuclei are located in like different areas. So they kind of appear to be stratified if you look at it, but then they're actually not. There's only a single layer of cells. And uh, if you have a stratified layer of cells, the way you can distinguish between how you can classify it is depending upon the superficial cell layer. So if you look at it like stratified squamous epithelium is going to have flattened cells with flattened nuclei in the topmost layer. So that's how you can classify it as stratified simple squamous epithelium. The same works for cuboidal and columnar. Cuboidal has a distinct round shaped nucleus, whereas columnars have elongated nuclei. So let's talk about the simple epithelium. Does anyone know the classifications or like what more it is further divided into?
Simple stratified epithelium?
No, I'm asking for simple epithelium.
Are you asking for squamous, cuboidal, like that?
Yeah, exactly. Perfect. So simple squamous epithelium, like you can see in the diagram here, has like flattened cells, and it is further divided into your endothelium and your mesothelium. So the difference between these two, it is an important thing which you need to know, is that for endothelium, the nuclei protrude into the lumen. So this can be found in your blood and lymphatic vessels. You will be seeing the slides on that shortly in your histology classes. And whereas in mesothelium, it does not have protruding nuclei. And this is the classic example of this is going to be your serous membranes. Next one is going to be your simple cuboidal epithelium. As you can see in the picture, you can clearly see the round nuclei, which is a characteristic feature to identify this kind of an epithelium. And it is found in your choroid plexus, the anterior epithelium of the lens, and germinal epithelium of the ovary, etc. The examples for all of these epitheliums is going to be very important for your microtests, and as well as this is like general knowledge which you're going to need throughout your entire semester. The last classification is a simple columnar epithelium. As again, you can see in the picture, you can see the elongated nucleus, and it has like two further classifications to it. It is classified as brush border and kinocilia. So brush border is found in the enterocytes, whereas kilocilia is found in the uterine tubes. Let's talk about the pseudostratified epithelium. So it is also known as a transitional epithelium, and pseudostratified cuboidal one I'm talking about right now. Sorry, my bad. And according to the shape of the superficial cells, you can identify if it is pseudostratified or not. And all the cells are anchored to the basal lamina. So no matter how long the cell is or how short the cell is, all of them will be anchored to the basal lamina. In the empty urinary bladder, you can see up to 8 rows of cell nuclei, and they are reduced to 2 to 4 rows of the cell nuclei in a full urinary bladder. This is why it is called as transitional epithelium, because it can transition depending upon how full the bladder is or how full it's not. So it can also be classified with, like without cilia, with cilia, and with sterocilia. So without cilia, it does not have any special characteristics. It's just plain, and it is going to be found in your prostate and ducts and glands. Now, with cilia, when we talk about it, you can see here in the diagram on the top, like little brush-like things. So that is going to be called as cilia. And the classic example of this is going to be a respiratory epithelium. This kind of epithelium like is present in all of the respiratory organs almost. Does anyone know why?
They help to brush the residue.
Exactly, yeah, for the mucus, for it to like flow and everything. And with sterocilia, so sterocilia, again, you can see on the top like little thread-like structures. Does anyone know the difference between cilia and sterocilia?
Stereocilia is not motile.
Exactly, perfect. So cilia helps in movements, whereas sterocilia does not help in any particular movement. So this kind of an epithelium with sterocilia is going to be found in a ductus epididymis and ductus deferens because it helps with like sperm to go through and like absorbs the nutrients and stuff. Let's talk about stratified epithelium. Does anyone know the further classification of the stratified epithelium?
Keratinized or non-keratinized?
Yes, but that comes under stratified squamous epithelium. What are the types of stratified epithelium are there?
Columnar and squamous again, no?
Yes, exactly. So, stratified cuboidal epithelium is also known as epithelium of transitional zones. This is completely different from your transitional epithelium. Uh, you can see it's called as epithelium of transitional zones because it can link epithelium of different structures. And this is found in the conjunctiva, part of the male urethra, and epiglottis. Stratified squamous epithelium is further divided into non-keratinized and keratinized. So in the non-keratinized part of it, you can see that there's flattened nuclei which occur in the most superficial layer. And this kind of epithelium, it covers most of our mucosal surfaces, such as your oral cavity, pharynx, esophagus, and so on. Whereas keratinized epithelium, it does not contain any cell nuclei, and it is found in your epidermis, which also means is your skin. So you can see it is further classified into thinly and thickly. So thinly is going to be found in almost all of your body surfaces, such as your face, your arms, legs, everything else. And the only exception to that is going to be your sole of the feet and palm of your skin, which is covered by a thick layer of keratin. Now, like Varun had mentioned earlier, basal lamina is going to be acting as your filter between the connective tissue and epithelium. So it's kind of like a barrier between the epithelium and your connective tissue. And the constituents of it are very important, which can be asked in the micro. As you can see here, they are collagen type 4, laminin, and heparin sulfate. It has two layers. They're called lamina lucida and lamina densa. And the examples of the location is going to be important again for your micros. And for that matter, even for your general knowledge, it is important because it can be asked in any of the questions when the professors ask you. The function of basal lamina filtration. So the basement membrane, it is like a vitreous homogeneous membrane, and the staining is kind of important here. The past stain purple red, that is important, which can be asked in your micro or for that matter anywhere else. And the location of it is pretty important. It is found in the kidney glomerulus and lung alveoli. Basal membrane is formed of either fusion of two basal laminas or a basal lamina and a reticular lamina. In the diagram in the bottom, you can see the reticular lamina. It's like a network, like a structure. And the function of this is like it helps in supporting, and it also acts as a barrier, and it is necessary for all the intercellular actions. That would be, for example, wound healing. Yes, and now Katrina will carry on.
Okay, just give me a second. And if anyone has any questions regarding that, that would be the perfect time. Um, okay. So can you guys see my screen?
Yes.
Okay, thank you.
Yes, we can. Yes.
Okay, so I'm going to be presenting glandular epithelium, and like my other colleagues already mentioned, these are things that you guys are going to see in almost every single slide throughout your year. So it's very good if you guys could understand this now. It's going to help you a lot, so you won't have to constantly keep coming back to this and having to relearn it. So what are glands? Glands are a cellular organ that will produce and secrete substances into either the bloodstream or into the outer surface. Examples are thyroid glands, sweat glands, mammary glands, and salivary glands. Now, it's okay if you guys don't understand a lot of these things in detail now, but try to at least understand what glandular epithelium is and how it works. So glandular epithelium is a type of epithelial tissue that forms the glands that are in our body. So they're covering the glands that are in our body. They are specialized. And because they are specialized, that means that they produce and secrete substances such as enzymes, hormones, sweat, and mucus. Now we have endocrine versus exocrine. Endocrine will secrete directly into the bloodstream. Exocrine will secrete through the surface to a duct. Again, endocrine, endo being in, it's going in our body, so it's going into our bloodstream, while exo means out, so we know that that's going to secrete to the surface through a duct. Most of the time, we're actually going to focus and look at exocrine because that's what we're going to see mostly in the tissues. So in exocrine secretory cells, the cells are going to synthesize a product, for example, sweat, and then they're going to release them as secretion. The following information is not as important, but just in case they ask, you guys also just keep in mind that they can also sometimes release what is known as incretions, which are just water and ions, or excretion, which are waste products. So I want you guys to look at this image because it's in your books, and try your best to at least visually memorize this, because unfortunately this is a part that you guys are not going to be able to see in your slides, but you will be asked about it in your credits and in your finals. So basically there are phases as to how this epithelium, how these glands secrete the substance. The first phase is going to be, give me a second. Right, ingestion. So in order for your glands to be able to make the products that they're going to secrete, these products are going to be built up or made up of other things. So first they need to uptake or ingest the things that they're going to use to make up this product. Then they're going to synthesize the product, and then there's going to be extrusion, where they're going to release the product either into the bloodstream, endocrine, or to the surface through a duct. And the best way to visualize it is actually the image, again, that's found in your books. You guys are going to start from the bottom and just go through the top. So you have ingestion, then you're going to have the synthesis, and then you're going to have extrusion or exocytosis. So just try your best to memorize how this image looks like. Now, modes of extrusion. Guys, this is something that is very important, so just please pay attention to this and really try your best to understand this, because this is something that you will most definitely be asked about, at least in your first slide test. So there are 3 modes of extrusion. The first one is ecrine. And basically, imagine the cell, right? So the cell has created, let's say, sweat, has created the sweat. When they release the product, when they release the sweat, the shape of the cell does not change. And examples of this would be like pancreas, salivary glands, or goblet cells. Apocrine is when the secretory product is discharged with the apical cytoplasm. Basically, the apical part of the cell, the top part of the cell, is going to be released with the product, and that means that the shape of the cell will change. In this case, the cell is going to become smaller than it actually was. For example, if the cell or if the epithelium is cuboidal and you see that all of the apical part of the cell disappeared, it is now going to look squamous. And this is seen in large aromatic apocrine sweat glands. Holocrine is the third method, and in holocrine cells, the entire cell basically just disrupts or just becomes damaged or explodes. The entire cell becomes a part of its secretion. And an example of this is a sebaceous skin gland, which you guys will learn about more later. But basically, once again, remember, there are 3 modes of extrusion. Eccrine, apocrine, and holocrine. The first one, eccrine, does not change. The cell shape remains the same. The second one, apocrine, the top of the cell disappears, so the cell becomes smaller. And the third one, the cell completely disintegrates. Now, based on these three images that I have down here, I want you guys to try and guess. So basically these images, the two middle ones are just one. They're all representing a mode of extrusion. So for this first one, I want you guys to guess which mode of extrusion is this. If anyone can tell me.
Eccrine.
Eccrine. Exactly. The little,
Eccrine.
exactly. So the blue circles are the product. And the product is being released and the shape of the cell is not changing at all. So who can tell me what the middle ones are?
Apocrine.
Exactly. So in here, you guys see this is the apical. So technically this is the apical part of the cell. It's extending. And the secretory products are here and they're being discharged together with the apical cytoplasm. So as you guys can see, the cell has become lower. The shape has changed. And then obviously the third one is holocrine. The entire cell has been disrupted. It's just pretty much ceases to exist. Now, we're going to classify the exocrine glands. And this is going to maybe seem a bit complicated, but just try your best to understand some of the terms that are going to be used, like uni, multi, exo. This will help you understand exactly what's going on. So when we classify them, we can classify them as unicellular and multicellular. And then we also have multicellular exoepithelial. But unicellular, basically, we're talking about single glandular cells that are inserted in a layer. They're in a layer of epithelial cells. And these are known as endoepithelial glands. Multicellular are, as you guys have probably guessed it, multiple layers. And we have multicellular exoepithelial, which consists of a secretory portion and the duct. And you guys will be able to visualize this better in a second. In multicellular exoepithelial, you can have unistratified, which is when you have the secretory portion, and this is formed by a single layer of cells, and this is going to be found in every single gland except for one. So if you guys are being asked, like maybe just a trick question or something about how to classify the specific gland, know that for the secretory portion of all the glands, they're all going to be unistratified except for one, which is going to be the sebaceous gland. The sebaceous gland is pluristratified, so you're going to see multiple cells in the sebaceous gland. So we can also classify them according to shape. So first we have tubular. It just looks like a tubule. You guys, I will show you guys images of this later in another slide, but you have tubular, and you have tubular, alveolar, and tubuloalveolar. And for each of them, you can have simple, branched, and compound, but for tubuloalveolar, it's just branched and compound. So remember, tubuloalveolar, either simple, branched, or compound, and then tubuloalveolar is just branched or compound. So for tubular, we have tubular simple, and a tubular simple will have one secretory portion and one duct. For branched, you're going to have multiple secretory portions that go into one duct. And for compound, the glands are going to have a lot of secretory units, and it's just going to be like branched, basically. Branched system of ducts. For alveolar, simple is not really found in humans, but just keep in mind that it does exist. Branched is when you have several acini into one single duct. And compound, the glands similar. They just have a lot of secretory units and a branched system of ducts. And then you have tubuloalveolar, which you have branched, which is again the same, multiple secretory units into one duct, and compound, which is the same again, a lot of secretory units and a system of ducts. So I know that all seemed a bit complicated, which is why we're going to try and break it down together. So here we have tubular and we have alveolar, right? This is how a simple tubular looks like. It looks like an actual tube. And this is what a simple alveoli looks like. And these are the secretory secretory portions. So here is where they're making the actual product. Branched, as you guys can see, well, it looks like an actual branch. Try to imagine the branches of a tree. Now we have like multiple. Same thing with alveoli. Here was one, but when it's branched, you guys see multiple of the secretory portions. One, and they're all connected to a duct, which you guys can't see here right now. Compound, here we have one simple, one of this is branched, and this entire thing is a compound. Same thing with the alveoli. Here we have a simple, here we have branched, here we have a compound. If I were to look at only one of these on the slide, you would see it as simple. A collection of them, you would see it as branched. This entire thing is compound. Thirdly is the tubulo-alveolar, which you guys know there's only branched and compound, which you guys can just, you're not going to be able to really see it like this, but just keep in mind that tubulo-alveolar has branched and compound. Now, structure of ducts. The structure of the ducts, guys, just basically try to imagine a tree. You have the main duct. Sorry, hold on. Right. Let's try to start from the actual bottom here. You guys are going to have the main duct, right? This is the main duct. Then you're going to have what's known as a lobar duct, which would be like this one, a lobar duct. And then you're going to have the interlobar duct, which is what I'm pointing to. Once you get to the actual, like, how could I explain this? The end part of the trees, now we're going into the actual lobules. And the ducts that you guys are going to find here are known as the intralobular ducts. And in the intralobular ducts, you're going to have two, striated and intercalated. Striated is going to be simple columnar, and intercalated is going to be simple squamous to cuboidal. So imagine, guys, that we're going to have like the secretory portion. Let's see if you, the secretory portion. The secretory portion is going to be directly connected to the intercalated duct. So imagine I'm making sweat, right? I've made sweat. The sweat is first going to travel through the intercalated ducts that are going to be here. Then it's going to leave the it's still within a lobule, guys. It's within a lobule. So the sweat is going to go from the secretory portion to the intercalated ducts, from the intercalated ducts to the striated ducts. Then it's going to leave the lobule to go to the interlobar ducts, because this is one lobule, this is one lobule, this is one lobule, this is one lobule. And here we have the interlobular ducts going through each lobule. Then it's going to go to the main like lobar duct, and then it's going to go to the main duct. I hope that this made a bit of sense to you guys. I always tell people, just try to imagine it as a tree. Remember that the smallest ones are the intercalated and the striated ducts. You guys are not even going to be able to see the intercalated ducts in your slides. You will be able to see striated ducts. You maybe will be able to see interlobular ducts, but then these two you will not be able to see. You just need to know that in theory. As for the epithelium, again, intercalated, just know that it's going to be simple squamous to cuboidal. Striated ducts are simple columnar. The interlobular ducts are simple columnar to pseudostratified. Lobar duct, pseudostratified columnar, and main duct is stratified columnar. And I know that this seems like very confusing, but these are just going to be things that you guys will have to memorize. However, trust me that with time, because you guys will be seeing this every single histo class, you are going to just memorize it immediately, and you will know that striated ducts are simple columnar, intercalated, simple squamous, interlobular, simple columnar, yada, yada, yada. And as for the bottom image, okay, no, this one is not in your book, but just another way that you can visualize it. This is like the main pancreatic duct, interlobular duct, then you have the intercalated duct, and here we have the secretory portion. And it's traveling. Okay. So now we're going to go to the secretory portion. Hold on. Okay, perfect. So guys, you guys are mostly just going to see three main types of secretory portions. The first type is the serous acini. Serous acini is pyramidal shaped. It's basophilic. It has a spherical nucleus in the basal third of the cell. I know this description sounds very weird and just like weird information, but in your credit tests, these are the things that they want to hear you guys say. Like they need to hear you guys say the word pyramidal shaped. They need to hear you guys say basophilic, and they are absolutely going to want you guys to say that the nucleus of Cirrus acini is in the basal third of the cell. The other secretory portion that you're going to see is mucus tubuli. Mucus tubuli is columnar. It has a light cytoplasm, a dark nucleus shape, and the nucleus is located at the base of the cell. At the base of the cell. You also have seromucous glands, and these are a mixture which I will talk about in a second. But before, I have these two images here. Just let's focus on Cirrus acini and mucus tubuli. We said that Cirrus acini was basophilic. The spherical nucleus is in the basal third of the cell. Mucus tubuli is columnar, light cytoplasm. The nucleus is located at the base of the cell. Out of these two here, which one do you guys think this is? Which secretory portion? Is this Cirrus acini or mucus tubuli?
Acini?
Yes, this is acini, guys. Basophilic, it's dark. You see the nucleus is 1/3 of the cell, in the basal third of the cell. In mucus tubuli, this is columnar. Look at this, it's columnar, and the dark nucleus is at the base, the actual base of the cell. So guys, these are secretory portions. So this Cirrus acini and this mucus tubuli, they're going to make certain products, and then they're going to send these products through the ducts out into wherever they need to go, either to the surface or to the bloodstream. They're going to send them out. So I know a lot of students tend to mix the ducts with the secretory portions. Remember, the secretory portions, which are these, Cirrus acini and mucus tubuli, they secrete things, they make products, and they're going to send these things through the ducts out of the tissue into wherever it needs to go. So please do not mix up ducts and secretory portion. Two different things. Oh, and these are known as seromucous glands. They're like a mixture of the mucus and the Cirrus acini. As you guys can see, like mucus, and at the base you have the Cirrus acini. This part is known as crescents of Gionati, and you guys will see that properly later when you guys study on your own in your histo books or in the lectures. Now that we talked about the different parts that constitute like the gland, which is the secretory portion, and the ducts, we're going to talk about the actual glands that you guys are going to see in your histo classes. Sweat glands. And I'm pretty sure this is going to come up in your first credit too. So there are three types. You have the eccrine glands, the apocrine glands, and you have the sebaceous glands. Can someone remind me really fast what this, by eccrine gland, what constitutes the mode of extrusion? What's going to happen to the shape of this gland? Is it going to change?
Doesn't
It stays the same. It stays the same.
It stays the same.
It won't
It stays
change.
the same.
It won't
Exactly.
change.
So again, sweat glands, you guys are going to have three. Eccrine glands, you instantly, when you see that word, you know that this gland, when it's making the sweat and when it releases the sweat, the shape of this cell will not change. So eccrine glands are simple tubular glands. Their secretory portion, remember, glands have the secretory portion and the duct portion. Their secretory portion is going to be light and it's going to be simple columnar. Their duct portion is going to be 2 layers of cuboidal epithelium. And eccrine glands, you can find them in your skin, except for your lips, glans, penis, labia. Okay, you guys will study those little details later. The apocrine glands, remind me again the mode of extrusion for apocrine glands. What happens?
So
The
it
shape
becomes smaller.
changes of the cell, but the cell doesn't die.
Yeah, the entire cell like disintegrates. Oh, sorry about that. No, it's the apical part. Yeah, the apical parts. Another secret to remember that the apocrine loses the apical is exactly this api. The apocrine glands will lose their apical part. So apocrine glands, they're branched tubular. The secretory portion is going to be simple cuboidal to columnar epithelium, and their duct portion is going to be 2 layers of cuboidal epithelium. Just as a way for you guys to kind of memorize it, if you guys are lost and you don't remember and they ask you the epithelium of a duct, guess most times it's going to be 2 layers of cuboidal epithelium. And here are the places where you can find epicrine glands. This is external acoustic meatus, nasala, axilla, etc. And then you have your sebaceous glands. And these sebaceous glands, they're holocrine. You guys know holocrine. It's going to completely disintegrate. And the secretory portion will be stratified, and so will be the duct portion. And this is found everywhere in your skin except in the palms of your hands and feet. Does anyone know what product the sebaceous glands secrete? It's in the name.
Sebum.
Sebum?
Yeah, sebum, exactly. It secretes sebum. And here is just an image of the 3 types. So let's look at eccrine glands, right? Here we're seeing 2 layers of cuboidal epithelium. So what am I looking at? Am I looking at a secretory portion or am I looking at the duct?
Duct.
Exactly.
The secretory portion.
No,
Oh.
we're looking at the duct. Yes, we're looking at the duct. 2 layers of cuboidal epithelium. I know that as for my own experience, I struggled a lot differentiating between the ducts and the secretory portions. One thing about histology is that sometimes you just need to memorize how things look. Sometimes you just need to memorize how things look. This is how the duct portion looks for eccrine. This is how the duct portion looks for apocrine. As you guys can see, this was the original. This is the secretory part. This is how the original part looks. And here is where they've probably released the product, because as you guys can see, there's a difference in the height of the cells. These cells are taller, these cells are smaller because they've lost their apical part. This is another good example. If you see something like this where half of the cell is tall and the other half is small, this is an apocrine, secretory portion of an apocrine gland. And then this is the sebaceous gland. And guys, remember this entire thing is the sebaceous gland. So this is the secretory portion, the light, remember, sebaceous, its secretory portion is light. And then we have the ducts, which you don't really properly see, but just know that it's pluristratified. And like I said, you're just going to have to memorize how these things look like. That's really the best way to learn it. And yeah, then we have the mock test, and I think I'm just gonna let Ria take over.
I'll be sending the mock test link to you in the chat right now, and you can go ahead with it.
And also, we're open for any questions. If you guys have any questions about anything, just let us know.
I have a question.
You have a question too.
So in our lecture notes from the Moodle, it says sebaceous gland instead of sebaceous gland. Is there like a difference?
It says sebaceous.
Yes, I distinctly remember reading sebaceous gland.
Okay, give me a moment just to double check. We've never learned of sebaceous. I'm a 4th year, so this was 4 years ago. I'm not sure if they added a new one, but
it's the sebaceous gland, but it's just spelled differently. Could that just be Professor Mokri spelling it differently, or?
I believe so. I believe it could also be a typo, like just a mistake.
No, it's spelled sebaceous in several places.
Oh, okay. Then perhaps it might just be give me a second. It might just be a new way of sebaceous glands. So I don't see anything online, to be honest. But if you guys just should ask him in class just to make sure. We only learned of sebaceous glands. I have never heard of sebaceous glands, but just make a note of it and ask him. He's an extremely nice professor. He's a very good professor. Oh, ask him anything you want. He will help you. But I
Thank
can't
you.
answer that. Yeah.
Yeah, I have a question.
Mm-hmm.
I don't know, I can't remember exactly, but it said something about the sebaceous gland being like unstrutted, something I can't remember.
Being on
I think it said unstratified or something. I'm not sure exactly.
pluristratified. Give me a second. Pluristratified. Can you guys see the screen? Yeah, so it has, see how here this only has one layer.
Okay.
Yeah, this is the really dark circle is the nucleus. So this entire thing is the cell, and you're only seeing one layer. In the sebaceous gland, you're seeing multiple layers, and it's pluristratified.
Okay.
Yes. Yeah. Just give me another. Yeah, I think this also no, not this part. Yeah, this also is what I think we're probably asking. That all glands except for one, the secretory portion, is unistratified.
Oh yes, that was it. Unistratified.
Yes, so all of the secretory portion for all of the glands is unistratified. For example, we were looking at these secretory portions, right? So the serous acinine, the mucus tubuli. These are all unistratified. You only see one layer of cells. Most of the times for all glands, the secretory portion is going to be unistratified, but there's only one gland where that is not the case. There's only one gland where the secretory portion is not unistratified. It's pluristratified, and that is the sebaceous gland. Because the sebaceous gland, the light part is the secretory portion, and as you guys can see, it has multiple layers.
Well, and pluristratified is the word you call it?
Yes, pluristratified.
It sounded made up, so okay. Thank you.
Does anyone have any other questions? Unfortunately, we do not have any specific microtest questions, but the mock tests which we have prepared have like basically summarized the kind of questions which can be asked in the microtest. And as well, I'm pretty sure you guys had a mock test in this week. So that's the only thing which we can offer you at the moment. But in the presentation right now, we did highlight some questions which can be asked in the microtest. So you could focus on those topics for now.
Someone said there's sebaceous gland instead of sebaceous gland. I just saw the lecture and I can't find it. If you can, so here's your lecture. It says sebaceous here, and then it says sebaceous somewhere down as well. Wait, it says sebaceous here. So I think this was a lecture we studied from, and you guys have the same one. So I don't know.
Yes, this is the one we have. I believe it was just a spelling mistake.
Yeah.
Okay. Someone's asked, are the PowerPoints enough to study? They are supposed to be the thing which you study from the most, like your first go-to resource when you study for the micros. But apart from that, the handbook you get as well, even that is very useful for you guys to study from.
Guys, one secret that will help you guys in the histology final a lot is to study for the micros and the credits through from the lectures. Because for the final, you will 100% need to use the lectures. I have personally not known of anyone who passed the final just using the handbook. And what happens is throughout the year, most students only use the handbook. And then once finals come, they open the lectures and they've never seen it before. So they're like, what? This is too much. And they can't study because they're overwhelmed. But if you start studying from the lectures now, even if it's just reading through it one time, once finals come, you know that there's like 30 pages. You're used to it. You've mentally prepared yourself to study that much content and you're going to be fine. So just read the lectures, use the lectures. The lectures are the best thing for you guys for histology, 100%.
The difference between using the lectures and the handbook is that the handbook mostly focuses on what you see the structures in the slide, which is important too, but then the lectures have got a lot of theory behind it, and which is very important in like the slide test, for example, because they ask you the specific details and the specific theory, which is not mentioned anywhere in the handbooks. So both of these resources are different and useful in their own ways. So yeah, but lectures you cannot ignore them. All right, does anyone have any other question? We can wrap up the meeting otherwise. All right, okay then. Thank you so much, everyone, for attending our seminar, and we hope to see you soon for the next ones as well.
Thank you very much.
Thank you.
Thank you very much.
Thank you.
Thank you.
Thank you.